The webinar will begin shortly.
Welcome, everybody. My name's Ken Foreman. I'm going to be moderating the program today. This is the third in our series of environmental science webinars. I want to remind you that all audience members are now muted and with your video turned off, but we would like this to be a conversation. So please, if you would like to submit questions, you can do so using the Q&A button at the bottom of your screen. The first of the two webinars in this series focused on the oceans and the coasts. Today's talk will explore life in desert crusts, which provide some insights into how plants adapted from the aquatic environments to land. MBL has a long tradition of convening students from around the nation to Woods Hole and linking research with education. The SES exemplifies this. For the past 22 years. The program has engaged undergraduates from more than 60 colleges and universities around the country in an intensive hands-on program focused on the study of ecosystems and global change. Students in the program spend about 20 hours a week in the lab in the field, and they cap their semester by designing and conducting independent research on questions that are of interest to them and to their mentors. Most of the work is here on Cape Cod, but many of the topics studied bear on questions being investigated as part of the larger research portfolio of scientists and faculty here at the MBL. Nearly 3/4 of the students that come through the program go on to careers in environmental research and education, and today you will hear from two recent alums of the SES. Jordan Stark, who attended the program from Skidmore College in 2013, and Hannah Gershon, who attended from Mount Holyoke in 2016. So after graduating from Skidmore, Jordan worked as a lab and field assistant in a number of projects, ranging from studies of plant traits at the Rocky Mountain Biological Lab to studies of prairie restoration in the Western Washington State and investigations of tropical soils at the El Verde field station in Puerto Rico, which is part of the Luquillo LTER long-term ecological research program. So after all of that, she returned to SES as a TA in 2017 and then transitioned into Zoe and Elena's lab, the presenters today, where she contributed to their studies of invasive algae and plant-soil microbial interactions. She is currently in graduate school pursuing a master's degree at Syracuse University. Hannah graduated from Mount Holyoke in 2019 and is now a graduate student at the University of Oregon. And some of the findings you'll hear about today arose from her independent research during the SES, which contributed to a publication in the Journal of Cell Science, in which she is a co-author. She is interested in all things botanical and has completed a number of internships, including as a botany field assistant intern for the Massachusetts Division of Fisheries and Wildlife, as a horticultural intern at the Minnesota Landscape Arboretum, and as a researcher in Arizona's Saguaro National Park as part of their Mosaics in Science program, where she studied the effect of encroaching development on soundscapes. I'd like to take a minute to tell you a little about today's speakers. Zoe Carden is senior scientist at the Ecosystem Center. She is an ecosystems ecologist, has been elected a fellow of the Ecological Society of America, and started her career working on the details of plant physiology. During her doctoral work at Stanford University, she studied the biochemical and cellular controls on stomatal function. Her explorations into plant biology have led her on an ever-broadening journey into studies of ecosystems and the microbiome. And her academic career path has included time as a Global Change Distinguished Postdoctoral Fellow at Berkeley, and faculty appointments at Bowdoin College, University of Connecticut, where she headed up the Biology Honors Program and was Associate Director of the Center for Integrative Geosciences, and finally here at the MBL. Within each of these very different institutions, Zoe has worked to build an interdisciplinary community supporting education and research essential for understanding and sustaining Earth's life support systems. Elena Peredo is a research scientist at the MBL Ecosystem Center with expertise in genomics and plant biology. She is broadly interested in the genetic basis of physiological traits that evolve under different environmental conditions upon colonization of new environments. She has worked on taxa ranging from hops to ferns to macroalgae and microbes. She received her doctorate at the University of Oviedo in Spain and was then awarded a Fulbright Fellowship, which took her to the University of Connecticut in 2010. She came to the Marine Biological Lab in 2012 as a postdoctoral researcher, was promoted to research scientist in 2018. In addition to the work she'll discuss today, her current projects include studies on the effects of the microbiome on success of invasive red algae in coastal ecosystems. So now I'd like to turn this over to Zoe. Take it away.
Thank you, Ken. I'll share my screen. All right, I'm very happy to be here today to talk to you about arid ecosystems and their future and the death-defying organisms that live in them. First, for context, I want to point out that, of course, you all know that the Western United States is a lot drier than the Eastern United States. But over the next century, we're expecting the Western US to become even drier. Whether we're looking at high emission scenarios or low emission scenarios for modeling, there's going to be a change in the percent soil moisture out there in the Western US and also even in the breadbasket states of between anywhere from -5 to 10 or 15% change in the soil moisture, loss of soil moisture. And it's also expected and actually already observed that we're going to see an increase in really heavy precipitation. The proportion of rainfall that falls in really heavy rainfall events and not as much in between. And we've already seen that in the northeastern US up here by Cape Cod, where we run the semester in environmental science, but also it's observed in the western US, where just a little bit more, 5% more of the rainfall is going to be in more intense storms. So that means that for this western area that's quite arid and where we have already 4 major deserts, we know that it's going to become even drier. Rainfall is going to become more sporadic, and we need to understand what that means for the function of both natural and agricultural systems and the organisms that live in them. So of course, if you're out there around Albuquerque, New Mexico, this is the landscape that you see. This is a natural laboratory already for understanding how plants cope with what is really a pretty harsh environment. There are a few of them there. You can see these little bushes around and about. And if you were to turn around 180 degrees, you would see this ridge that is covered with this carpet of stuff, rough looking stuff. That is microbiotic crust. It's a crust made of microorganisms that bind themselves and the soils together. So yeah, there are a few higher plants, but it's mostly this crust. And you can see these stripes along here. That's where bicycles have ridden in the past. These crusts are quite fragile, and they take from decades to centuries to recover once damaged. If you zoomed in along the path, you'd see this crust is really a cohesive unit. And you would also, if you were walking in the Western US, in national parks, you would see signs like this: don't bust the crust, stay on the trails, protect the fragile soil crust. And that's because they're really important for ecosystem function out there. They are the dominant on a lot of areas of the landscape. They're the dominant organisms there, and they can collect nitrogen from the atmosphere, fix it into forms that's available to other organisms. They can photosynthesize extensively. They can control water infiltration, and they can control the soil that is being stabilized against windstorms and those more intense rainfalls that we expect. But of course, it's really fascinating what's in those crusts. These literally are microbes, and some of them are little green balls, 5 to 10 microns in size, that are very similar to the organisms that are inside higher plant leaves, to the cells that are inside higher plant leaves, I should say. So they share the same biochemistry, the same photosynthetic capabilities, a lot of the same genetic programming. And yet these guys living out there in the crust are able to be fried by 50 degrees C temperatures. They're able to withstand long periods of drought and then use rainfall really rapidly when it's available. And they're even being hit with very high UV light loads. So those extremes would kill most plants, and do in our gardens, even our house plants. But these tiny cells, for whatever reason, are able to survive. How? Well, we're interested in them because they're green algae and cyanobacteria. There are mosses, there are lichens out there. It's quite diverse. And heterotrophs also are within those crusts, feeding on those organisms. There's a whole food web. Now, I don't want you to think that these are only in the Western US. If we go back to Massachusetts, here's the Woods Hole, where a semester in environmental science is. If you go up here to the parabolic dunes at Cape Cod National Seashore, it looks like this. You see these big dunes, and in between the grasses, again, is some crust. This black stuff between these grasses is made of mostly actually green algae. And if you pick up a little bit of sand, it doesn't just fall apart, it can balance on the end of your finger because it's bound together by these microbes. So going back out west, I first became interested in these microbes. I was introduced to them by Louise Lewis about 20 years ago, and she's fascinated by the transition of green plants or algae from water to land, originally way back in evolutionary history. And her idea was that by studying these very resilient organisms that are in the crusts, that are single-celled, maybe we would learn something about how the first algae made that leap to land. A first step for her and her colleagues was simply to do a survey of what green algae are out there. Are they really diverse, or are they just one type that's everywhere? And it turned out, using molecular methods that they found, that in fact, they are incredibly diverse. They had to use molecular methods because the green algae are just these little green balls, hard to tell apart. They sequenced the 18S ribosomal RNA gene, put together an evolutionary tree. You don't have to see the details here. It's just showing how all these different kinds of organisms are related across a bunch of different classes of green algae. And at all of these little blue dots, that's where there's some desert alga that was detected, that was identified. So they're very diverse. But even within a particular group, within a clade of green algae, within the Cynodesmaceae, a family, you can have both aquatic organisms and desert organisms that are very closely related. They share genetic background, yet somehow desert ones are able to survive, drying out, heating up, and the aquatic ones are not. Why is that? Well, it's a beautiful study system for studying how desert and aquatic species are different. And we can do it via several mechanisms. Been doing this for about 20 years. A very recent paper summarizing some of it is at this Journal of Cell Science publication that Ken already mentioned. But let's just look at some cell biology and a little physiology, and then in the next portion, Elena will talk about gene expression. So first, cell biology. Among all these aquatic and desert taxa, interestingly, aquatic ones tend to be multicellular. We don't exactly know why, might have to do with predation pressure, but they tend to be multicellular, and when they divide, they pop out of the mother cell, and there they all are stuck together. This is under a bright field microscope. If we look with a confocal microscope at some desert algae that are dividing, you see individuals coming out of the mother cells. They're not stuck together. And you see that this red stuff here, that's actually the chloroplast. That's fluorescence from the chlorophyll in the chloroplast. That's really handy for tracking cells, but it's also really handy for looking at the physiology of these organisms, because chlorophyll fluorescence is a very useful way to probe non-destructively how active these organisms are photosynthetically, how much photosynthesis they're doing, and how efficient it is. So you can put them, grow them on little glass beads, and you can put them underneath this fiber optic, and you're monitoring this fluorescence coming off of them, just like you did under the microscope. And you can tell from that what they are doing as they dry down and wet back up. And this is what Hannah Gershon contributed to in our work. And very briefly, some of her results are shown here. Up on the top, we have a graph from an aquatic alga and then two desert ones down below. On the y-axis, we're just looking at a measure of photosynthetic activity or efficiency. And here you can see this first bar is when all of those cells have been dried down. So if this were completely dry and we were looking at it, and there's very little photosynthetic activity. You add a raindrop and the desert algae pop right back to life. Within seconds, they begin photosynthesizing. The aquatic one, nope, dead as a doornail. Again and again, you can cycle this over and over. The same algae dried down and wet up will pop back to life over and over if they are derived from desert environments. So we're going to hear from Hannah next, and then after that we'll move into a discussion of the gene expression that seems to underlie this incredible ability of these plants, green algae, to deal with this harsh environment. Hannah?
Yeah, thanks. So, hi, I'm Hannah. I'm a 2016 SES alum. As Zoe already mentioned, for my final research project, I studied the desiccation tolerance in aquatic and desert algae with Zoe and Elena, who are both really wonderful mentors. I think that the final research project in the SES program is really unique in that it gives you the opportunity to completely immerse yourself in a research project that both you and your mentors care about, and that you're involved with every step of the way. I think that the SES program makes you work really hard, but as I've heard other SES alums say, it's all really fun and it's all work that you care about. Your fellow students in your cohort, your TAs, the faculty, the scientists are all really care about you and want to help you succeed. I remember my cohort and I, while working on the research projects, would all come together at least once or twice a week and work on our final projects together. I think that's another really wonderful thing about SES is that it's so interdisciplinary. It brings together students from all different departments, backgrounds, specialties into one program. So for example, if I didn't quite understand the stats equation I needed for my research, I could reach across the table and ask John, who was a stats major, to help walk me through it. So not only do you get to work on a research project you really care about, but there's this sense of community and collaboration that you get throughout the whole program. I think that during the SES program, I realized that I love learning about the science. Like I love getting knee deep in the salt marsh. I realized that that research wasn't really the career I wanted to follow, and that I was more interested in scientific education. So right now I'm actually a master's student at the University of Oregon studying environmental education and the environmental humanities and how they can help us reframe social justice issues and move towards solutions. So yeah, I would highly recommend the SES program. It's great. And I'll hand it off to Alina now.
So hello. It's been great to see you again, Hannah. It was such a lot of fun to have you in the lab. So yeah, she probably remembers me screaming around when we see the algae just going up and down and reviving one time after the other. As I said, we care about these projects a lot, and I've been studying this algae for a long time with Zoe. And I'm gonna go back to one of her slides here. So see, am I sharing now? No, I'm not. So I'm gonna share my screen, sorry. And I'm gonna pull back this slide from her presentation in which she walked us through how important these microbiotic crusts are, the many roles they perform in an ecosystem, and what are the conditions these organisms have to endure. So they have to endure streams of temperature, water livability, and light. So how they survive there, that's the question that I really care about. And they survive because they are desiccation tolerant. These algae have an extraordinary physiology. They have adapted their cell biology. And of course, this is controlled by changes in gene expression. I am working, and Zoe and I, we've been working extensively in this family, the Synodesmaceae, that she talked a little before. And that is because it provides us with a unique opportunity to study aquatic and desert species that are extremely closely related. So if we look into this tree here, we can see that it's a very diverse family. There are more species than those represented here in the tree. And if we look to the bar of substitutions, that indicates how closely related these species are. When we go to the branches, we see that they are tiny. And this is because they are really, really closely related. And in just this tiny phylogenetic space, we have 3 independent leaps to land. And for each of these leaps to land, we know of distant, closely related aquatic relatives. And this is important because it allows us to ask questions in a very powerful way. So we can get the question of how do desert evolved algae survive in harsh conditions of deserts, and use this comparative approach to rephrase the question in what is that the desert evolved algae do that aquatics do not. This is, again, it's a unique opportunity that green microalgae give us in research. So also, these green microalgae they're actually quite relatively easy to grow in the lab. And that, again, is important because we can move our experiments from the field into the lab and use a lab-based experimental approach that allow us to work with single isolate, that is a single species of algae, under very controlled conditions. So we can grow the algae in liquid culture, and we can control how they grow, and we can control how we desiccate them to study their different responses. So basically, one of the approaches we use is to transfer these algae that are happily growing in liquid media under almost ideal conditions with the amount of light they like, the nutrients they like, and we can place them in tiny tiny pieces of glass where we're going to control how we dry them down. And we do that by controlling the humidity in a chamber. And this way, we can repeat this process multiple times. And we can repeat this process with multiple species. And we can do it exactly the same way every time. So we can compare the responses of it across different taxa. So usually, if you get these tiny drops of culture and you place them over a counter, they're going to dry in something between 10 to 30 minutes. By controlling the evaporation rate, we allow them to desiccate over 12 hours. And this is important because it allows us to identify different stages when they desiccate. And also, we control for other variables when we're working on a lab. We try to isolate the variable we want to study, in this case, water loss, from other environmental variables that could affect what is happening on the cells. So we study desiccation under dark conditions, but still we monitor very carefully what is happening with each of our experimental units, each of these tiny dots over here. And to do so, we use an astronomical camera that basically allows us to see us almost in the dark. So now I'm going to play a tiny video in which we're going to see each of these dots to dry down over that 12-hour period. So here goes. You can see the dots in the dark drying and drying. At around 12 hours, they dry down. And we do this, so we monitor that everything is happening the same way for each experimental unit we are working with. So we identify five stages that we consider important for desiccation. Of course, the first one would have to be that in which the cells are fully hydrated and they have never been exposed to stress. So the volume that we accounted at that point was 100% of the droplet of algal culture that we were studying. And then through the process, through the desiccation time course, we focus in that stage in which they only retain 80% of the volume, 40% of the volume, that is a late desiccation state. They're growing in media. So when they have lost 60% of the original volume, that implies that also the salts in the media have concentrated. So that's also important to keep in mind when they're dry. And also we wanted to study how they recover. So we also study what happens once they have recovered, once we added water and we allowed them to recover for an hour. So how we study what is happening, how do we study the changes in gene expression over this period? So what we did is we extracted the DNA from the cells, and we used the KEC facility here at the MBL to sequence using Illumina sequencing the mRNA of these cells. We used a variety of bioinformatic approaches, quite a lot of them, to try to understand what was that the cells were doing, what were the changes in the gene expression that were associated with these different stages of the desiccation and rehydration time course. So now we're going to see some graphs. So these are heat maps, and each of them corresponds to a desert species. So we look to the changes in expression in the desert species, and what we're visualizing here is the overall summary of what happened. So here what we have basically is a representation of those genes that have been impacted by the time course of desiccation and rehydration. So in each of these graphs, a column represents a sample, and for each sample corresponds to a stage. So we have duplicate samples for the hydrated state, for the early desiccation, for the late desiccation, from when they're dry and when they're rehydrated. In each of the rows, we represent the fate of one gene through the desiccation and rehydration process. So if a gene starts in golden, it means, and finishes in purple, it means that it started through the process, it downregulated its expression levels went down. While on the other case, if they started purple and went golden, it means that they upregulated their expression. So we can see that in both of the desert species, there was a huge impact of desiccation and rehydration in their expression patterns. So here we summarize the expression patterns of almost 2,000 genes. We also cluster them to better study how is that they were responding. So here in these graphs, we represent the log-fold change in expression. That is how many times they change the expression. And we have clustered here. Each of these groups corresponds to one of the graphs. We cluster how they behave through time. So there is one group of genes that is downregulated, another that responds with a moderate upregulation, and another that responds with extremely upregulation of the gene expression. And you can see that the most extreme point always corresponds to the dry stage. Okay, so they respond, but what is that they're doing? We knew from the literature that there were certain, or there are certain genes that are affected by water loss in plants. So there has been some work done in seed plants using as model system resurrection plants, that is plants that are able to survive desiccation. They do so by activating a series of strategies. So to survive desiccation, and that is you will be worried if your houseplants look like this, but actually these guys will recover completely just after a few days you add water. So to be able to do this, what they do is they activate the antioxidant production, they increase the accumulation of sugar, They accommodate proteins, the type of heads-up proteins and late abundant proteins. They increase the strategies that leads to photoprotection, and they downregulate the photosynthesis. So basically where they are completing is a series of strategies that allows them to better prepare for that water loss. So we look to the very same genes that are upregulated in seed plants. And we saw that in these desert evolved green microalgae, they were behaving in a very similar way. So in these graphs, in the y-axis, what you have is the fold change in expression in red, that is the amount of change. And you have the gene abundance, that is the amount of transcript in the gray boxes. In the x-axis, what we have is the different stages. The dry stage is indicated in red. And we can see that genes that traditionally have been identified in seed plants, they're also present in green microalgae, which this group of algae separated from seed plants 500 million years ago. So this indicates that these strategies are extremely shared and conserved across the green plant clade, which goes back to the question that Zoe remind us that was one of the reasons to study this algae. How do plants conquer land? How do plants move to land? Are these strategies conserved? Why do we have to study organisms beyond seed plants? So yes, the same genes are present and they respond to water loss in seed plants that they do in green microalgae. And those processes involved with photoprotection, antioxidant production, or accumulation of head-sock proteins and Lea proteins are affected. What happened with the aquatic? So we know the aquatic won't survive. We know it dies. We know it can't tolerate water loss. So what is that it's doing? So when we look to the overall expression profiles, what we see is that actually they have a ton of changes. There are a lot of genes here in golden that are upregulated through the process during water loss. And if we look into which these genes are, in fact, there are many of the same genes we're seeing in the desert species. Again, those involved with photoprotection, those involved in antioxidant protection, and those involved in osmotic protection are upregulated. They are present in their genomes and they respond to water loss. And yet, these cells don't survive water loss. So what is that is happening? As I said before, there are many, many genes that are impacted by water loss, and we cannot visualize one by one 2,000 genes. So what we use is an approach that allows us to cluster genes according to the function and study what is happening with that function specifically. So we did that, and we identified a series of functions that were impacted in all three species, independently of whether they were the desert evolved species or aquatic ones. So here in these graphs, there is a lot of information. So I'm going to walk you through it. So in this inner circle here, each of these tiny sectors here represent one function. So for example, this function here represents parts of the chloroplast. And you can see that it's purple. And it's purple because overall, the genes that control this function are downregulated in this desert evolved species. So in this outer circle, what we did was to plot individual genes that had been annotated within this category of functional behavior of genes. And we can see that although there are some genes that are upregulated, most of them are downregulated. So overall, the category, it has been downregulated. So there are some cases where most of them are downregulated. So here, one of the categories involved in chlorophyll binding, most of the genes are downregulated in both desert evolved species. In fact, when we looked to the desert evolved species, very few gene categories were upregulated. So some of them involved in the membrane behavior. And this is very important because membranes are deeply affected by water loss. So you need to change the composition of membranes to better survive water loss. Also, another category that was impacted were the ones involved with the oxidative stress. And these were, in both desert evolved species that we studied, the only categories that were affected during desiccation. Overall, in all the other gene categories, what we saw was quite a drastic downregulation, and that affected functions involved with energy and lipid synthesis, again, with chlorophyll binding and this photosynthesis, many of the different aspects of photosynthetic activity were downregulated. That's what happened with the aquatic then. So when we look to the aquatic, what we saw was that yes, those categories that were upregulated in the terrestrial species were upregulated, but all the other ones had also response to water loss with an upregulation. So they had increased their metabolic expended in energy. They have also increased photosynthetic activity over here. And this is, it's not the way that you better prepare for stress. So way of expressing what the aquatic is doing is, if you just think of what happened when your computer doesn't really do what you want to do, what you want it to do, and you just start pressing different keys. And instead of making things better, actually what you do is your computer to get even more and more stuck. Well, you know, somebody that knows better will just tell you, well, you just need to press escape. And that will be enough to get your computer back in order. So in a way, that is what the aquatic is doing. If we go back to the original question, what is that the desert-evolved algae do that aquatic do not? What we know now is not just affecting those very key essential genes for desiccation, those that has been traditionally associated with protection functions. We know that those genes are there. We know they respond to water loss, but it's not that. What is happening in this group of algae is that the desert species are responding in a calm, very orderly manner. They are activating the protective strategies and downregulated the rest of the metabolism, ready to survive the stress. While our little aquatic friends, they are just panicking all around, and they are trying to do everything at the same time without really responding to the stress that they are going under, but just panicking. So this is what we really need to do now, is to look how generalized this response is, how this is encoded, and how it's controlling the genomes of these species. And that it just illustrates how powerful all those organisms that have evolved in different conditions are to study different stress and different adaptation processes. So now I'm going to pass the torch to Jordan. Here we have Jordan with some of the algae that's isolated from desert crust. With this, I'm going to stop sharing. I'm going to turn my camera off. And Jordan, you're on.
Hi, thanks, Elena. So I'm Jordan Stark. As Ken said at the beginning, I was an SES student back in 2013. And then went back and graduated from undergrad. And after that, sort of bounced around to several different positions as a field and lab tech in different places, which was kind of an adventure. And it was really incredible. In every single one of those positions, there was something that I took on, something that I was able to do because I learned it in SES. It's really, when I think back on the SES program and what I got out of it, that sort of breadth of understanding. Every week there's a new topic, there's new techniques that range all the way from field work to lab work to then getting some data and putting it together into some kind of analysis by the end of the week. And that was really different than what I got at my undergrad institution. It's just so immersive. Definitely was the most challenging semester of my undergrad experience, but I think in a good way. And as Hannah said, it's with that community of other students and all the people who work at the MBL. But all the students are doing the same work together, and so there's a lot of good collaboration that goes on there. I came back, I went back to Woods Hole in 2017 as a teaching assistant for the SES program, which was also a very cool experience to sort of see it from the other side, getting to see students sort of come in and learn all of these different skill sets, practice all these different techniques, and then apply them in these independent projects that they take all the way from start to finish, and really grow as scientists. And then I worked as a research assistant in Zoe's lab with Zoe and Elena, which was also fantastic. I worked on tons of different projects, including some work related to this that we've heard about today, and learned, again, lots and lots of different things that I've been able to take with me. I'm now a graduate student at Syracuse University. I work on microclimate and how it interacts with plant communities. So it's a project funded by the National Park Service. So I design and build new sensors and then go deploy them in the Great Smoky Mountains National Park. I've hopefully got about 100 sensors out there right now recording soil moisture and temperature. And then I'm bringing all that data back and trying to make models of microclimate and the plants that are associated with particular microclimates. So really the skills that I learned in SES and also just sort of the attitude towards science that I learned in SES of you know, trying different techniques and working through the inevitable challenges that come up, I think has served me well ever since. So if you're considering the program, I definitely recommend it. And I think Zoe has a little bit to finish up.
Yes, thank you very much. All right, well, I'm going to just share my slides one more time. Oops, sorry. And I hope that you've enjoyed learning about arid ecosystems and about their future, what we think their future will be in the United States, and also about these incredible death-defying organisms that live within them. And because one of the major emphases in the semester in environmental science is looking forward and trying to understand what's going to be happening to our planet, and because I'm a total photosynthesis nerd, I wanted to end with this. The fluorescence that we were able to use even under the microscope to see individual cells, and they were able to use to probe photosynthesis, is actually even visible from satellites, from space. And there's this wonderful quote from Buckminster Fuller that says that there's just an outstandingly important fact regarding this spaceship, this ball that we're on out there floating around in space. There's no instruction book. We need to learn what the knobs are, what the responses are, and how all the systems interact. And so one of the frontiers in ecosystem science at this time, right now in 2020, is the deployment of satellites that are circling the Earth, and they are detecting tiny little chlorophyll fluorescent signals and telling us about what's happening during the seasons in South America, North America, the distribution of photosynthesis and productivity in agricultural natural ecosystems planet-wide. So with that, Ken, I'll send it back to you.
Okay, well, thank you very much, Elena and Zoe. And now we would like to open the seminar up for questions. So I want to start with a question, actually not so much a question, but a comment that comes from Amy Townsend Small. Amy is a professor at the University of Cincinnati, and she writes, "Hi, Jordan. I was an SES student in 1997, the first cohort. And I also went to Skidmore. Nice to meet you."
Small world.
So I also have a question here from someone watching who says, "Do you have any estimates of the decrease in net primary production due to projected decreases in SM? SM, what's SM? Do you know, Zoe?
Soil moisture.
Soil moisture, thank you. So obviously this is a soil scientist writing, and how will that affect food webs and chains?
Oh, well, that's a huge question. There are estimates, there are modeling estimates of it. I'm sorry, I don't have the numbers right off the top of my head, but as you can imagine, if you're decreasing the western, the moisture in those western systems by 15%, and it's already pretty low, that's a pretty dramatic shift. But you might also say, well, they're already pretty dry. So making it even drier maybe isn't wiping out all that much productivity. So it's a really interesting balance of trying to understand where the organisms are in those ecosystems, what their sensitivity is to the change in the amount of moisture available and what that means. We have, of course, been seeing horrible wildfire fires in California, and we know that there have been some really dramatic droughts in the breadbasket area of the United States. So we do already know that there are visible events occurring that are of concern. But definitely from the point of view of modeling, there are estimates, and I would point people to the either the IPCC Information International Panel on Climate Change, or also one might go to some of the US climate assessments that are being done that have really interesting resources online.
Okay. So here's a cell biology question. It makes a lot of sense, probably for Elena, maybe. It makes a lot of sense that the genes involved in membrane remodeling are upregulated during desiccation. Is anything known about the specificity of these changes in plants? For example, is the plasma membrane recomposition differently than, say, chloroplast membranes?
That's a great question. And actually, I don't know if we know that there are differences between the cytoplasmic membrane and the chloroplast membrane in composition, how they change individually. What we do know is that there are severe changes in the composition of them, and also differences in the way that the chloroplast will behave. I've seen in the chat that Thomas has another question related to oxidative stress and photosynthesis. That's yet another question. Could you, if you don't mind, because I really don't know how the membranes composition change, and actually I don't recall reading much about that or anything. Could you read the other one, the oxidative stress one?
It makes a lot of sense that, let's see, that's the finding that desert algae can...
that's the first, yeah, finding, yeah.
Can almost instantly regain photosynthetic capability upon rehydration was interesting. I was under the impression many desiccation-tolerant plants systematically shut down photosynthesis to reduce generation of, so ROS is oxidative stress.
So
Elena, can you help me with ROS during
so
drying?
reactive oxygen species.
Ah, okay, thank you.
Okay.
So is this, the question is this not the case for algae? How conserved do you think the mechanisms are of desiccation tolerance throughout the plant kingdom?
So I'm gonna show what happens with the oxidative stress in this algae when they're dry. And then I think so I can elaborate on why it's so fascinating that they regain photosynthetic activity so fast. So so I'm gonna share my screen for a sec here. So oxidative stress is the accumulation of can be caused for the accumulation of reactive oxygen species inside a cell when the cell cannot process, for example, the electrons that come from the photosynthetic apparatus. So here, what we did is we actually visualized the accumulation of oxidative stress. We have our aquatic species here on the left. We have the desert evolved species on the right. And we use fluorescence, a combination of fluorescent to visualize what was happening. So we can see the cells of the algae because we're seeing their chlorophyll signal represented in red. And then we use a dye that is activated when there are reactive oxygen species present inside the cell. So these are unstressed, fully hydrated cells. Then we dry them down, and we rehydrated them with this dye to visualize what was happening. And if we look to the terrestrial species, to the desert evolved species, they are certain species like Baja California, because that look like nothing has happened. There is no signal at all of accumulation of reactive oxygen species. In some others, we might see some, especially in the cells that are dividing. So here in yellow is represented that accumulation with the dye. In other species, again, in the pyrenoid area and a tiny bit on the cytoplasm, we can see a tiny accumulation of reactive oxygen species. So whatever they're doing is preventing them to accumulate this kind of stress. Well, if we look to the aquatic ones, they look like Christmas trees under the microscopes. They have significantly increase in the amount of reactive oxygen species that have been accumulated on the cells. Of course, because the cell membrane integrity probably has been compromised, we can see it in the periplasmic space here in red, in yellow. This is the space between the cell wall and the membranes that we were talking about before. So they are really effective controlling the amount of stress and the amount and the accumulation of reactive oxygen species, which contributes to this extraordinary physiology that Zoe can talk about now.
Okay, so I have a couple of questions that ask about tardigrades. So tardigrades are certainly not plants, but they do have the ability to be desiccated and to revive. So this one is a wonderful presentation, especially hearing about the paths of students. Has anyone collected tardigrades at the same sites and you know, studied how they desiccate and revive? I'm not aware of any SES projects on that, but Zoe, I wonder, Zoe, if you think there's any, you know, similarity in terms of the cellular mechanisms.
Well, first, in response to the question, I don't know of people who collected tardigrades as part of that group. However, it was a National Science Foundation biotic crust surveys grant that supported it. So there was interest in the diversity of organisms that are out there, but tardigrades in particular, I don't know. But at the NBL, there are people who are really interested in desiccation, for example, of rotifers. And we have talked about putting together projects and grant proposals that compare some of the strategies that are in these algae and strategies among rotifers. I think you could add tardigrades into that mix. But there are some parallels probably. For example, LEA proteins, these LEA proteins, they may well be something that's quite similar across all kinds of groups that are able to withstand desiccation. But going back to one of the questions before, where there was a question about whether the desiccation tolerance in these green algae and the strategies within them, the circuitry within them is exactly identical to what might be found, say, in resurrection plants, which are larger plants that can be dried out. It's not exactly the same. And from the point of view of photosynthesis, there's something that has been lost among higher plants and during evolution, and we don't know why. And that something is that during the dry down, the drying of these individual green algal cells, whether they're out there in a microbiotic crust or whether they're in lichens that dry out a lot and are found all around the world, during that drying, there is some protective mechanism that kicks in that siphons the energy captured by photosystem 2's light harvesting complex, just siphons that energy away from photosystem 2 and protects it. So it actually pulls the energy away, and photosystem 2 essentially is blind to that excess excitation energy. We don't know what that mechanism is exactly. There are various ideas in the literature on what it is, but it's a really fascinating protective function that we do not see in higher plants. It's not the xanthophyll cycle. Whoever you are who asked that, you know what it is. It's not the xanthophyll cycle, but it's something sneaky, something beautifully sneaky that is happening in these cells.
Great. So here's a question from a prospective SES student. And if Jordan's here, Jordan, if you could answer this, she asks, how do you choose your independent research project or topic while at SES? Do you have full capability of choosing what you want your research project to be on? Since you were a TA, I thought you could give some insights.
Yeah, I can take a stab at that, and then you can correct me if I get anything wrong. I guess the way that the program is structured through, I don't know, the first 2/3 of the program or so, every morning or almost every morning, there's a lecture from a researcher. That might be a little bit like the lecture today, where someone's talking about what they're working on, or it might be sort of a more basic science topic. And so a lot of students find particular lectures or particular research that's going on at the Ecosystem Center really grabs them, and they would like to do something that's related. And so they'll talk to those specific scientists about working in their labs for an independent project. So usually it's something that sort of develops between a student's ideas and something that's already going on, or that there's already sort of capability to do at the MBL. But there's a huge amount of flexibility, and the range of projects that happen is pretty incredible, from work with animals to work with nutrients and plants and all kinds of different ecosystems.
Hannah, any thoughts on that? Are you there still?
Yeah, I would just echo Jordan in saying that like Zoe, I remember Zoe's lecture when I was a student talked about photosynthesis, and that really grabbed me, and led me to work with her and Elena and like other students that had the same paths. But the research projects are really fun and collaborative. Yeah.
Okay, and one last question for Zoe, I think. Does this work provide any insights on how to make crop plants use water more efficiently or become more tolerant of desiccation?
Yeah, that's a really good question. And of course, it's one of the things that drives our interest in this, because as I said during the talk, a lot of the genetic circuitry and a lot of the physiology that's in these tiny little individual green plant cells is really similar to what's in the individual cells within higher plant leaves. So yeah, there's a large research community. It's not just us. There's a large community of people who are very interested in what makes some resurrection plants, larger plants, desiccation tolerant, what makes these little cells desiccation tolerant, the genes that are expressed, the genes that are wound down during desiccation and rehydration. And there are efforts even to transfer genes. There's genetic engineering that's being done to transfer protective genes like those that Elena was talking about, to amplify their numbers or transfer them from a particularly desiccation or drought tolerant species into another kind of species to see whether that can help improve the drought tolerance of the desiccation tolerance of a particular type of plant, a corn plant, for example. I think another thing that's of real interest is people are realizing that over time, as we have bred plants just through selection, we've bred crop plants to be very productive above ground and make a whole lot of fruit or grain or whatever it is that we're interested in. That has sometimes, and in fact often, led to a change below ground where we're not allocating, the plants are not allocating as much effort, as much biomass down below ground. They're not necessarily taking it, using the soil resources as efficiently as their ancestors did before the breeding. So there's a whole nother group of people who are really interested in understanding what those ancestors of our current crop plants were actually doing out there in landscapes, some of them quite arid landscapes like those that we've talked about today. And so that kind of information, even if there are just hints of it still embedded in the genetic machinery of our crop plants, could be something to build on.
Well, thank you very much. And I think it's now 12:58, so we're going to wrap this up. But I want to thank everyone who tuned in today, and I want to thank the panelists for taking time to make this presentation, and remind you all that the next one of these webinars will be December 1st. It will be on human impacts in the global nitrogen cycle and nitrogen in the coastal zone, the good, the bad, and the ugly. And it will feature Jim Galloway, who is University of Virginia, but also a trustee of the MBL and a frequent visitor to the Ecosystem Center, as well as Javier Loret and myself, talking about work that we're doing in the coastal zone. So I hope you'll make an effort to tune into that one. And thank you all very much for coming today.